Okay, picture this. You're looking through a microscope. On your left, you've got a perfectly healthy human cell. On your right, a rapidly dividing, completely rogue cancer cell. Now, their DNA blueprints are basically identical, so we've got to ask the big biological question here. What changed? To actually figure this out, we can't just stare at the static DNA. We need to see what it's doing. Think of it like comparing a perfectly orchestrated symphony to absolute chaos. 1 minute, a gene is playing its part. The next, it's blasting at maximum volume, and crucial tumor suppressors totally silenced. So how do we actually capture that massive shift? Welcome to The Explainer. Today, we're diving deep into the RNA sequencing pipeline. We're going to track the journey of a single sample from the physical wet lab straight through the computational dry lab and see exactly how raw molecules are transformed into literal, life-saving biological discoveries.
Here is our roadmap for the process. First, we'll look at the gene expression mystery. Then, from cells to sequencing. After that, alignment and quality control. Then, normalization and gene quantification. And finally, differential expression and discovery.
Let's start with part 1, the gene expression mystery.
So, in the old days, we relied on tech like microarrays, which, only let us look at genes we already knew existed. But RNA-Seq, it completely unconstrains the whole process. We're talking about a massive dynamic range here. It means we can spot entirely novel features, identify really dangerous gene fusions, and even catch alternative splice sites without needing a reference genome to guide us. It is literally the ultimate tool for decoding cellular behavior.
Moving right along to part 2, from cells to sequencing.
This is the wet lab phase. First up, we've got to physically extract that incredibly fragile messenger RNA from our cells. Because it's so delicate, we use an enzyme to reverse transcribe it into much more stable complementary DNA, or cDNA. Once we have that, we ligate or attach specific sequencing adapters to the 5' and 3' ends, kind of like snapping tiny little barcodes onto each fragment. Finally, we amplify the whole library and load it onto a glass flow cell. And just like that, the physical prep is done.
Now the chemistry kicks in. Inside a massive, high-throughput machine, like the NovaSeq 6000, that flow cell undergoes sequencing by synthesis. As new DNA strands are being built, fluorescently labeled nucleotides give off these unique flashes of light. The cameras inside the machine capture millions of these tiny color signals cycle after cycle. It's honestly pretty wild to think about.
Which brings us to part 3, alignment and quality control.
This right here is the crucial handoff. All those millions of microscopic chemical flashes of light, they get computationally translated into raw digital text data. Specifically, they become what's known as a FASTQ file. So now, we've got millions of short text sequences called reads, which represent pieces of our original genes. But at this exact moment, it's really just a massive, unorganized pile of genetic letters.
To actually make sense of all those scattered reads, we have to map them back to the human genome. But there's a huge biological catch here. Our reads are from mature mRNA, meaning the junk DNA, the introns, has already been spliced out. If we just try a straight alignment, it completely fails. So we use computational algorithms, like STAR, to perform what's called a spliced alignment. This smartly skips right over those massive intron gaps, utilizing the conserved donor and acceptor splice sites to reconstruct the true, active structure of our genes.
Up next, part 4, normalization and gene quantification.
Okay, so once everything is aligned, we count the reads. But hold on. If gene A gets 500 reads and gene B gets 10,000, we can't actually trust those raw numbers yet. Why? Because longer genes just naturally generate more fragments. We absolutely have to normalize the data. If you're comparing different genes within the exact same sample, you'd use a within-sample method, like TPM. But remember our original question? To compare our healthy cell against our cancer cell, we need between-sample normalization, like TMM. That corrects for outliers and different RNA composition, so we know our cancer signals are actually biologically real and not just weird technical artifacts.
Finally, part 5, differential expression and discovery.
How do we actually see these discoveries? We use a volcano plot generated by differential expression tools like DISC2. This scatter plot literally erupts with data. On the x-axis, you've got the magnitude of change, essentially how drastically the expression shifted, measured in log2 fold change. On the y-axis is your statistical significance, or your p-value.
Those dots shooting way up into the top left and right corners, those are our culprits. Those are the highly significant, drastically altered genes that are actually driving the cancer. And look, this isn't just some abstract computational exercise. This pipeline literally saves lives. Researchers are out there using it right now to discover targetable cancer biomarkers and to screen exactly how tumors react to drugs at a molecular level. It even scales up for single-cell RNA-Seq to give us high-resolution oncology data, or to profile incredibly complex microbiome metatranscriptomes in the gut. We started with a total mystery inside a microscopic cell and navigated through chemistry, optics, and some seriously complex algorithms to get our answer. The core takeaway: mRNA goes in and actionable biological discovery comes out. But think about this. As this pipeline gets faster and even more precise, if we can decode the errors in a single cancer cell today, what currently incurable diseases will we finally be able to silence tomorrow?